How's it going, class? This is going to be the second of the bonus lectures that I'm going to be doing for this course. This one's going to focus on the bioenergetics that we briefly talked about. I know I kind of talked about this a decent amount in the first part of our second lecture, I believe. I just wanted to go over it a little bit more and in more detail. I think this material is really, really important for understanding where energy goes, what we do with our macronutrients, and just kind of understanding how these are utilized specifically in sport. Previously in my assessments course that I teach, it's a fourth level course. I've had a lot of students come in, they don't know really anything about these bioenergetic systems, and they don't know anything about really where the ATP is coming from and what these things mean and how do we utilize them. So I want to go over them in more detail just so I can know that we are understanding these concepts, right? So we'll jump in here. All right, so we'll talk about our bioenergetics. Bioenergetics is just a word broken into two, right? Biology, energetics, right? It is how we are getting our energy from the biological processes within our body. So in this case, ATP production. How are we getting our ATP? And we know, or we should know by now, that there is three energetic systems. So the first one is the phosphagen system, also known as the ATP PCR system. Um, this system is anaerobic. It's going to be our fastest source of ATP because it's a very simple reaction. We'll get a little bit more into each of these in detail after I go through the slide. Um, the glycolytic system is another anaerobic system, and if you remember, anaerobic means without oxygen, so it has no oxygen, and we're not utilizing oxygen for this reaction whatsoever. So second fastest system. And then we have our oxidative system, which is our aerobic system, right? We can use this to break down fats. We can use this to break down carbohydrates. It's our slowest source of ATP, but it is very complex and methodical, so it allows us to have the most efficient ATP production from our substrates, our macronutrients. So the first one here is the phosphocreatine system. Right? Like I said, short duration, high intensity. This is something that you would use for sprinting, weightlifting, and jumping. I make the analogy, I think I might have already made it in the lecture, to Usain Bolt. So Usain Bolt, fastest man in the world, runs the 100-meter dash. So when he gets a decent amount of time into his race, he gets 60, 70 meters into his race, you begin to see his speed drop off. And that might be because he's pounding his chest and pointing to the crowd because he's already won, because he always wins, right? But a good portion of that is due to the phosphocreatine system being depleted of its substrates. So since it's beginning to run out of this phosphocreatine here, it's pen up here, since it's beginning to run out of this phosphocreatine here, it is harder for us to resynthesize ADP into ADP or ATP, right? So since there's less phosphocreatine, that means that this process right here, this resynthesis of ADP into ATP, is less likely to occur. And if it's less likely to occur, it's occurring more slowly. That means that there's not as much energy for the body, which means that you can't have as much muscle contraction, you can't have as much force output, and you can't move as fast. Remembering that this is anaerobic, so it does not require oxygen. No process or nothing in this biochemical process requires oxygen whatsoever. In highly anaerobic animals, you'll actually see that this system is more developed. So in animals like the cheetah, which spends a predominant amount of its hunting time waiting and then sprinting, you will see it have a much more highly developed anaerobic system for developing ATP than, let's say, some sort of animal like probably a whale, right, other mammal, right, that spends a large portion of its time just doing long transits over long distances within the water. ATP is stored in the cell, PCR is stored in the cell, and this ATP is broken down into ADPs, right? We know that this ATP is broken apart. It gives us the energy, and that's how we create all of our bioenergetic process. That's how we create all of our ATP. And so we look at this reaction here. The reason that it can occur so quickly and the reason that it's so simple and gives us that massive explosion of energy, remember I make the analogy to a firecracker, that quick explosion of energy, is because all that needs to happen is this phosphocreatine right here outside of the mitochondria, in the cytosol, is donating its phosphate group to ADP, and we're making ATP. And that's one quick reaction that requires one enzyme, cytosol creatine kinase, right? That's all this is. And a fun fact here for those of you who are interested in muscle damaging, creatine kinase is something that is located within the cell. So it's located only within the cell because its process is to do this. It is to rephosphorylate ADP into ATP. When you go through bouts of exercise and you get muscular damage, you get rupture of the cell membrane. And when you have the rupture of the cell membrane, you will get creatine kinase leaking into the systemic circuit. And that creatine kinase in the systemic circuit is actually a way of measuring or quantifying objective muscle damage after a certain period of time. And now that's different for elite versus novice trained individuals. But it is a cool thing to know in that things within the cell are actually released when you get muscle damage, and we can measure those to guess the amount, or not guess, to indirectly quantify the amount of actual muscle damage you had in certain areas. And so going back to this here, this says all creatine kinase, all it does is one simple reaction and boom, we get ATP really, really quick, right? Snaps through it really, really quick. But we also burn through that phosphocreatine really quick. And so burning through it very quickly. This system has a maximal capacity up to about 10-ish seconds. And again, that can be changed or augmented over time with training. Usain Bolt, the same example I used before, his phosphocreatine system is probably active much more efficiently and much more long term than somebody, say, is a marathon runner, just because he's trained that system, he's stressed it, so it's adapted, and it's become more improved over time. Now we look at anaerobic glycolysis. And anaerobic glycolysis, as it says, is another energy production without oxygen. It's our moderate duration, moderate intensity energy system. Now, I don't really like that it says moderate intensity. This is still for pretty high intensity. You can maintain a pretty high intensity for up to about a minute. So I would say moderate to high intensity is a little bit more accurate. It says up to about a minute. That can be anywhere from a minute to up to 2 minutes if you train the system properly. So for those races like the 400-meter dash, the 800-meter dash, if any of you were in track, you know that the 400-meter dash is considered to be the most brutal run in all of track and field, just because you are essentially sprinting for the entirety of the race. So it is very taxing. You're getting a massive amount of acidosis, metabolite accumulation, as well as PCR depletion while you're doing that. And it's said to be very, very tough. And we know that anaerobic glycolysis is producing ATP. But more importantly, right, we see how little ATP it produces. It only produces 2 ATP molecules. What's more important is the pyruvate molecules that it is producing. The pyruvate molecules, and we'll ignore lactate for now, ignore this lactate for now. The pyruvate molecules are what are being shuttled into the mitochondria to then be used by the oxidative system via the Krebs cycle and the electron transport chain. So if we use glucose, we'll get 2 ATP molecules. And if we use intramuscular glycogen, we will net 3 ATP molecules. Let's see if I can explain that better to you here. Right, essentially, what is going on is when we use glucose, we have an input of 1 ATP here. We have an input of 1 ATP here. And then we have an output of 2 ATPs and an output of 2 ATPs. So we have -1, +2, which nets us 2 ATP molecules. Well, when we have a glycogen, this step right here, this phosphorylation of the glucose to make it glucose 6-phosphate, when we use glycogen, this step is not needed. So we don't actually use this step because glucose, while it is in the glycogen molecule, is actually stored as glucose 6-phosphate. So this top part here is not needed. So now if we start from glycogen, which would be starting right here, we have -1, +2, which nets us +3 like we see here. So those of you who saw that and are maybe a little bit confused, I wanted to explain that in a little bit more detail. It's not super important for this. We're usually going to talk about just this glycolytic process from glucose itself rather than from glycogen, just in case you guys were wondering on that one, right? This occurs in the working muscle. So this is going to be ramped up depending on how hard your working muscle is being activated. So if you're sitting here right now listening to me, you are probably not heavily using this energy system because it's not very strenuous. You know, maybe the amount of times that I say the word right in my lecture, I've noticed that that's my filler word. I use the word right all the time. I started listening to a couple of my lectures, the later ones, and it just makes me want to bash my head against the wall. So I apologize for that. I'm doing my best get rid of those. I know it's annoying, but I'm doing my best that I can to get rid of that word. But but as you sit here, you're not really utilizing this energy system. So the rate of this energy system producing ATP is rather low because it's not very stimulated. We use the glycogen in the muscle as well as we draw glucose from the blood to use for this energy system. And this occurs in there's a typo here, does not occur in the cytoplasmic reticulum. It occurs in the cytoplasm of the cell. It may occur in the cytoplasmic reticulum a little bit, but we use specifically, it occurs in the cytoplasm or the cytosol. They're relatively the same thing. And about 2,000 kilocalories of glycogen stored in the liver and the muscles, as well as stored glycogen is not easily moved from distant muscle to working muscles. So really what you have is, let's say you're working your legs, what you have to rely on is the stored glycogen within your legs. Let's say you're doing leg extensions to make it more specific. You have to rely on the stored glycogen within your quadriceps muscles for the leg extensions, as well as you're relying a little bit on the blood glucose that's already circulating. And then as that exercise becomes more taxing and as it lasts longer, you're starting to rely on the released glycogen as glucose from the liver specifically. It's really hard to mobilize stored muscle glycogen from, let's say, the pecs or the arms and give that to the legs. Really, what you're doing is if you're running at a stored glycogen in the legs, you're mobilizing it from the liver only. So having more stored glycogen in your upper body isn't really helping you for lower body exercises in terms of glycogen storage. Now we're looking at this reaction again here, just so you can kind of see what's going on, see all these moving parts, as well as understand what's going on with these high energy electron carriers. So these NADHs. These are also really important because these are going to then go to the Krebs cycle to help us produce those are going to go to the electron transfer chain to help us produce ATP. So glycolysis produces ATP, produces pyruvate for the Krebs cycle. And we have our heavy work rates when we are working too fast. When this pyruvate is being produced too fast, let's say we're working out really hard, and this pyruvate is being produced too fast, so fast so that this pyruvate right here, sorry to skip around, this pyruvate right here cannot be shuttled into the Krebs cycle fast enough. We're working so hard. Glycolysis is running so fast. We're producing tons and tons of this pyruvate. If this process is producing pyruvate too quickly for the Krebs cycle in the mitochondria to handle it, it sits around and it becomes lactate, which at the same time happens to be when acidosis is being produced, hence the name lactic acid. We talked about that before, but the accumulation of lactate is actually good because it helps to buffer that acidosis, and then it can be later utilized as a fuel. It can be sent to the liver to go through gluconeogenesis, or it can be reconverted into pyruvate to be then shuttled back into the Krebs cycle. And so again, we're looking at this reaction here. It's the same thing written over and over and over again. Just so you guys can understand it. If you're a chemistry person, you want to look at the carbons, want to look at where the carbons are going, so you understand what's occurring, where the phosphates are going. But in essence, what we're saying is the glycolysis now is extremely important for both just the glycolytic system, but also the oxidative system, because fast oxidative work is done by breaking down glucose. And if we remember, slow oxidative work is done by breaking down fats. But in this case, we're still talking about glycolysis. Gives us pyruvate, which is shuttled into the Krebs cycle, which, as you can see here, only produces a small amount of ATP. Again, we're only getting a small amount of ATP from it. What really gives us our maximal amount of ATP is the electron transport chain, which is part of aerobic respiration or aerobic glycolysis. So aerobic glycolysis, also known as the oxidative system, again, we should know is a long duration, low intensity exercise energy system, or just for what you're using, let's say at rest. Usually you'll be breaking down fats if you were at rest, but if you're doing some sort of low intensity exercise, you'll probably predominantly be breaking down carbohydrates for that exercise bout. It begins with glycolysis. Specifically, we're talking about breaking down carbohydrates, begins with glycolysis. If we're breaking down fats, it begins with lipolysis. The Krebs cycle is the oxidation of pyruvate. The Krebs cycle, also known as the citric acid cycle. We see this redone here, pyruvic acid, pyruvate, same thing, broken down into acetyl-CoA, and then it goes to the Krebs cycle, and we get lots of these high energy electron carriers, as well as a little bit of ATP. And these high energy electron carriers are what are crucial for the electron transport chain. Okay. The electron transport chain uses a redox reaction and electrochemical gradient. We don't get into that too much, but it breaks down the hydrogen ions and Krebs byproducts. So it uses the NADHs and the FADHs to essentially create ATP. And so the one glucose molecule will net 32 ATP or 33 ATP. Why 33 ATP? Well, if we remember all the way back here, one glycogen molecule gives us 3 ATP. So glycolysis, or one glucose molecule, depending on if we're getting it from blood glucose or if we're getting it from intramuscular stored glycogen, will give us 32. Or if it's intramuscular stored glycogen, will give us 33. Because remember, we're skipping that initial step. So we're using one less ATP to begin the steps. So that means at the end, we are netting more. And this is specifically aerobic glycolysis for carbohydrates. Remember, there's difference between carbohydrates and fats. This is pretty much the same thing. This is just showing you the breakdowns. The one thing this does have added here is it shows us the electron transport chain down in this corner here. And if we remember, what we're talking about is what happens is these NADHs that were produced, right, this NADH here that was produced in the Krebs cycle, was produced by glycolysis, is coming here. It's essentially donating its hydrogen ion and it's creating a concentration gradient. And I'll try to draw this out without making you think that I'm a 4-year-old because I can't draw on here. Much like my handwriting. If you saw my handwriting, you'd think I was you would think I was holding the pencil in my mouth and try to write words. That's how bad my handwriting is. But the NADH comes in here, donates the hydrogen ions. Now we have lots of hydrogen ions in this space here. This is the intramembranal space within the mitochondria. Okay, so we have lots and lots of hydrogen ions up here. High concentration gradient. If we know anything about our concentration gradients, just like water, it wants to flow down its concentration gradient. So if there's more hydrogen ions up here than there is, right, than there is down here, hydrogen ions are going to want to flow this way. They want to flow down, which is what exactly is going to happen. This is what I talked about before. This is the only time in the body where you were actually turning mechanical energy into chemical energy. Usually chemical energy, like we're talking about here, everything we're talking about this class is chemical energy into mechanical energy, using ATP to move. That's chemical to mechanical. This, we're using mechanical to chemical. And what's literally occurring is that these hydrogen ions are moving through this ATP synthase, causing it to crank kind of like a water wheel. And it's cranking like a water wheel, which is causing energy to be initiated into this ADP in inorganic phosphate, giving us ATP. Now, the summation of all this is that you should understand that this is a very complex and long-term process. It takes a while. I mean, in terms of our daily living, it doesn't take very long at all. It only takes a few seconds or minutes. But in terms of the other bioenergetic processes, right, you can do a maximal jump like that. I can say, get up out of your chair and do a maximal jump real quick. You can get the phosphocreatine system to fire that ATP right now. But it's hard to get the glycolytic system and the aerobic system specifically to fire as quickly as that. And when you're breaking down fats, even more so, because breaking down those fats, as we should know, is even slower. So we look at our energy systems here. We have a mitochondrial fat, mitochondrial CO. So these would both be the oxidative system. This is just starting with lipolysis. This is starting with glycolysis right here. This is the glycolytic system, and this is the phosphagen system. So the average ATP turnover rate, how much ATP you can make per kilogram per second. As we see here, anaerobic glycolysis, ATP produced per fuel unit. So we see it is much less efficient, but its production rate is much faster, okay? Same we see here, the turnover rates. You see the turnover rate here for the phosphagen system is much, much faster. It produces ATP very, very quickly, but it is very inefficient. We remember that one PCR only gives us one ATP. So we burn through that substrate extremely quickly. And then finally, we look at aerobic glycolysis, a little bit slower production rate. And then finally, aerobic lipolysis is even the slowest production rate. But it produces the most ATP. So you can see these things are inversely related, right? The faster it produces ATP, the more quickly it burns out, and it can produce the ATP more quickly because it's the least complex. The more complex reactions take longer to produce ATP, but they pump out the most overall ATP because they are the most efficient. And I know I'm beating this dead horse here, but this seems to be something that is lost through literature, through people coming up when I talk to some of my friends that are athletic trainers, some of my friends that are coaches, they don't understand these energy systems at all and how they're applied to sport. And they're important to understand so you know what you're training, you know what you're utilizing, and you know how to augment or change them going forward. So finally, altogether, remember, all these energy systems are always active. ATP, PCR system, the phosphagen system, glycolysis, and the aerobic system are always on. They're never off at any one point. There's just a certain period in which they are the most efficient at doing their job. Even though the phosphagen ATP PCR system only lasts maximally for about 10 seconds, it's still always on producing a little bit of ATP. Glycolysis ramps up and then it ramps down and kind of keeps going, but slowly begins to diminish, but it never fully diminishes. And then aerobic kind of ramps up over time and then kind of maintains this steady state here over time as you continue to do exercise, right? So the stimulus is what drives the usage of these energy systems. If you have something that's really highly intense, you're going to use a phosphagen system. Something that's more moderate to high, you're going to use glycolysis. Something that's moderate, probably aerobic glycolysis. And then something that's really, really low intensity, you'll use aerobic lipolysis, just because it takes longer to break down those fats. And so you can take that time because it's more efficient. So again, phosphagen, quick energy, simple, boom. And then the glycolysis is kind of like burning newspaper. And then aerobic is kind of like burning a large log. It takes a long time to burn, but you get a lot of heat production from it and you get the most overall heat output. So that is going to be the end of this lecture here. Keep an eye as we go through. I'm going to be posting flyers for different research studies, as well as I'll be posting a flyer for my own research study. They're worth the same amount of points. Obviously, I would love for you to join my research study more than joining someone else's, but we want to see people in. We want to see people get involved in our studies. We have a lot of research studies going on right now, so the opportunities will be very rampant. I'll be posting those, as well as I'll be posting more of these bonus lectures going forward. Keep an eye on those things. If you guys are starting to have questions with the quizzes, starting to have questions with any of the content that I cover, make sure that you are reaching out to me through email and asking me questions specifically on what you want to know, as well as potentially scheduling meeting times with me so we can discuss these concepts. Also say now, make sure that you are staying up to date on this class, okay? These are hard concepts. The exam is not meant to be really, really hard, but it's not going to be easy. It's going to be asking you questions on concepts, things that you have to understand. If you were just going through and you were memorizing the material, if you're saying, okay, glycolysis produces 2 ATP, I guess that's a memorizable fact. But if you're memorizing that the phosphagen system is the shortest duration, it has the least complexity, it has the fastest production of ATP, it's the least efficient. If you're remembering all those things separately, it's going to be difficult for you. You have to conceptualize this course. You have to look at the concepts and be like, okay, what's going on here? If this is occurring, what's occurring down the line? What's occurring above the line? How are we changing these things? Okay. So make sure you're keeping up on these things because this is an upper level exercise physiology course. So you're going to need to stay on top of it because when you get to the end of the testing session and you have that test week, we'll do the review session in that first part of that week. But if you have not started studying yet, you're probably going to struggle on the exam. And there's only 2 exams. So if you struggle on the first one, you're going to have to work extra hard on the second one to get your grades back up. And I'm there. I'm here to help you. I want to talk with you and I want to help you understand these things. But I can only do as much as you guys let me. So that'll be the end of this lecture. And I'll see you guys in the next one.